Take out the refrigerated urine. Opening the cap. Take out 1 ml of this urine. In a centrifuge tube. Stop. Then we will centrifuge the urine at 10,000 rpm for 2 minutes. Stop. After centrifugation, a cell pellet will be obtained. Then we will add 1 more ml of urine to the cell pellet. We will again discard the supernatant and repeat this. And now repeat the process of centrifugation until 5 ml urine has been processed. Next we will add 250 microliter of the first lysis buffer, which is GDUR1, to the cell pellet. Vortex spin, okay. Followed by re-vortexing and incubation at 70 degrees Celsius for 10 minutes. Followed by incubation at 70 degrees Celsius for 10 minutes. Add 250 microliter of the second lysis buffer, which is GDUR2. Invert mixer tube 4 to 6 times. Next we will add 350 microliter of the third buffer, which is GDUR3. Invert mix again for 4 to 6 times and centrifuge again at 10,000 rpm for 10 minutes. Decant the supernatant into a DNA mini spin column provided with the kit. Centrifuge at 10,000 rpm for 30 seconds. Discard the flow through and insert the column again into the frit. Next we will add 600 microliter of membrane wash buffer to the column. Centrifuge again at 10,000 rpm for 30 seconds. We'll discard the flow through again. Microliter of the wash buffer again to the DNA spin column. We'll centrifuge again for another 30 seconds. Again. Sorry. Through again. Centrifuge the empty column again at 10,000 RPM for 1 minute. Transfer the column into a fresh 1.5 ml centrifuge tube. Stop. Take the nucleus free water, pipe it out 50 microliter of this nucleus free water and apply it evenly on the column. Incubate at room temperature for 1 to 2 minutes. Post incubation, centrifuge the column at 10,000 RPM for 30 seconds. After centrifugation, discard the column. Discard the column, and your elute contains the HPV DNA. We will store it at -20 degrees Celsius for future use in RT-PCR.